Hello everyone, this is Dr. Nita Srivastav and welcome to ITLS Academy. So, today I'm going to talk about product development, regulation and safety. And when talking about the product development, we have to discuss about the key factors that are responsible for the product developments, the regulation associated to the products, as well as the safety concerns of the product. So now talking about the development of a new product. So when development of any new fermentation product or any other industrial product, it depends upon the following factors, and those are market, current level of scientific knowledge and regulatory environment. So talking about the market. So market basically depends upon the product uniqueness, that how valuable the product is. And this market, it is obviously affected by product uniqueness and where a market is perceived to exist for a new product, and this can be referred to as a market pull. We can say it as the market pull. And for example, if I take the case of any medical product, so suppose a new drug is developed for the treatment of a previously untreatable disease, or we can say that a more effective drug is developed, then this can be profitable for a company at a higher extreme. Now, when a product is developed, so market size is also a major consideration. For example, if a drug is targeted, the development of a drug is targeted towards the rare disease, then it might not be economically viable for a company. So rare disease, if I talk about the rare disease means that it occurs in a very few people, like around 1 to 2 lakh of people. So basically the development of drug for these rare disease, they comes under the orphan drug programs in countries like USA, Japan and in European Union. But in India, if I talk about these orphan drugs, then the drugs which are associated with the rare disease, they are known as the orphan drug, and they comes under the orphan drug programs. In India, orphan drugs, actually they are defined in the New Drugs and Clinical Trial Rules 2019, and these drugs, they are intended to treat a condition which affects not more than 5 lakhs of persons in India. So if talking about the rare disease, there is no definition like rare diseases in India, but still it is covered under New Drugs and Clinical Trial Rules 2019. 2019. So this was about actually the market. Now talking about the current level of scientific knowledge. So as with the advancement in science, it has resulted in the rapid development of industrial processes, and we can say it as the scientific push. Like when we talk about the penicillin, so the Fleming's discovery of penicillin in 1928, it ultimately led to the establishment of the antibiotic industries in 1940s, and even a particular rapid development was seen with monoclonal antibodies, and their production was first developed in early 1970, which led to a multi-million dollar industry within just a few years. Further, The rapid progress of recombinant DNA technology also in recent years has generated a push for many new products. And importantly, we can say that this technology, it can be readily applied to established fermentation processes. So this was about the current level of scientific knowledge. Then talking about the regulatory environment. As we all know that when we talk about any pharmaceutical product for example, it actually takes around 10 to 12 years to come to a market, and with a lots of investment, a higher cost is invested. So now just to reduce this, the target is to reduce this time period from 10 to 12 years to 8 years. And a a lot of factors, they influence the long development time, and they are the regulatory environment, which involves safety of the product, employees, and the environment, along with the legal aspects, patents, product licenses, and social and ethical aspects. So these three important factors, they are involved for the development of any new product. So as I said that the journey of process from a lab scale research to a marketable new product, it takes a long time and it is a very costly process, and we can see that it generally takes, as I said, 10 to 12 years to come to the market. And the factors, they involve the regulatory environment and so on, and the target is to reduce this to a maximum of 8 years. Now the steps which are involved in the development of a new product. So new product development, it is the development of original products, product improvement, product modifications, and new brands through the R&D efforts. This process consists of following steps: idea generation, idea screening, concept development and testing, marketing strategy development, business analysis, product development, market testing, and finally the commercialization of the process and the product. So now, after talking about the development of a new product, now as we know that the development of a product, it is the result of hard work of many years, and therefore it is necessary to protect the invention, and the invention can only be protected if it fulfills the criteria of patent protection. So now first of all, we should understand the patents and their importance. So if I define patent, so patent is the granting of a property right by a sovereign authority to an inventor. An inventor is someone who invents invent some process or product. This grant, it provides the inventor exclusive rights to the patented process, design, or invention for a designated period in exchange for a comprehensive disclosure of the invention. So as I said that for a designated period. So normally it takes around 70 to 20 years depending upon the country. But if I talk about India, the Indian patents, then this period is around 20 years. So after the completion of the 20 years, the invention normally comes into the public domain. Now these patents, they can be separated into three distinct types. First one is the product patent, second is manufacturing process patents, and then methods of use patents. So now first of all, talking about the product patents. Product patents are for the substances, composition of matter and devices such as bioinsecticides, recombinant proteins, monoclonal antibodies, plasmids, etc. These are most readily protected as patent infringement can be determined by product analysis. Now talking about the manufacturing process patents, for example, DNA isolation, purification of a recombinant protein. Then talking about the method of use patents, it involves a novel role for a product such as mode of bioinsecticide application, drug delivery, etc. Now talking about our biological patent, what is a biological patent? So a biological patent, it is a patent on an invention in the field of biology which allows the patent holder to exclude others for making, using, selling, or importing the protected invention for a limited period of time. Now, this biological patent, it may include biological technology and products, genetically modified or microorganisms, and even genetic material. And the scope and reach of these patents, they vary among jurisdictions like Australia, Europe, India, China, US, etc. So now talking about the criteria for patentability. Actually, there are three criteria for patentability. First one is the novelty, second is non-obviousness, and then industrial applicability. Now talking about the novelty. Novelty means where the patent applications are assessed. It requires that the claims in the patent, it should be totally new. Means for an invention that was previously unknown and unavailable to the public when the patent application is filed. Now talking about the non-obviousness. Non-obviousness means the invention cannot be obvious to a person of ordinary skill in the field. Means the invention should show something surprising and unexpected in order to be non-obvious. Means it should not have some ordinary skill or some expertise in the area. So this is a very important criteria also. Now talking about the industrial applicability. So the industrial applicability means the invention, it should be useful. It should be useful and beneficial and should have some industrial applications for uses. Now talking about the patenting of microorganisms. So as per the provision of Patent Act in 1970, the inventions in the areas which were relevant to biotechnologies, they were excluded from patenting. For example, any method for agriculture or horticulture, any process for the medical, surgical, curative, prophylactic or other treatment of human beings, or any process for a similar treatment of animals and plants. So according to the Patent Act 1970, living entities like animals, plants or microorganisms, they cannot be protected by patents. However, however, the inventions relating to the processes for the production of substances like enzymes, antibiotics, alcohol, etc., or bioconversions, which were utilizing the microorganisms, they were patentable. So now, when we talk about the patenting of microorganisms, the patenting of microorganisms were excluded in the provision of the Patent Law 1970. But after the decision of the US Supreme Court in the case of Diamond versus Chakravarti, a patent protection was not granted to microorganisms as product claims. Means previously, before the decision of this case, microorganisms were not granted for a patent, and only the process claims in which microorganisms were used, they can be patented. So now the detail I will just discuss about this case. So a case of Chakravarti and Diamond versus Chakravarti in 1980s, it opened the gates for the patentability of microorganisms. So what happened? A claim of a microbiologist. His name was Dr. Ananda Chakravarti. So he filed a claim means for the grant of patent for a live human-made and genetically engineered bacterium, which was capable of breaking the component of crude oil. It was accepted by the US Supreme Court. Now, in this case, what happened? The controller of patents of the United States, he denied the claim for patenting the bacterium. How? Stating that microorganisms are product of nature, and anything that is the product of nature cannot be patented, and hence are non-patentable according to the US patents regime, which was reversed by the United States Courts of Customs and Patent Appeals. So now, dejected by the decision of the US Court of Customs and Patents Appeal, Sidney A. Diamond, who was the Commissioner of Patents and Trademarks, he appealed to the US Supreme Court, which again went in favor of Chakrabarti by establishing by establishing that a human-made, genetically engineered bacteria was capable of treating oil spills, and thus it was an invention that was accompanied by, just see, by novelty, usefulness, utility, non-obviousness, and industrial capability. So here we can see the different criteria that are required for any invention to be patented. So this invention, uh, it actually comprises of all these criteria like novelty, usefulness, utility, non-obviousness, and industrial applicability. Thus, by this case, it was proved that any microorganisms which was human made can be patented. So, and it this has a lot these criteria which are naturally occurring microorganisms was not capable of. So this was actually the case which opened the gates for the patentability of microorganisms. So since then, many living or modified microorganisms, they have been patented. For example, we say Oncomouse, it was the first patented mammal. So we can say that anything under the sun that is made by man is patentable. And we can just talk about the quick facts that after the completion of human project in 2003, nearly 25% of the genome, that is more than 4,000 genes, they are already covered by at least one US patent. And it also includes genes for the Alzheimer's disease, colon cancer, asthma, and in particular we can see BRCA1 and BRCA2 genes which are associated with breast cancer. and even since 1980s, genes considered to have been isolated from the natural state and purified, they have been eligible for patent protection. And the first few patents include the DNA altered for producing specific proteins like insulins. These are some quick facts that covers the patentability of the genetically modified microorganisms or the process. Now talking about the product quality and safety. So we can say that prior to approval for marketing any new product or food or healthcare product, it is to be used by humans and domestic or farm animals. It must be thoroughly tested, and this involves the aspects of safety and efficacy and the required specifications for purity and activity. And product licenses, they're granted only after the product has passed all the requirements of the regulatory system. So in the case of pharmaceuticals, we talk about the case of pharmaceuticals, the process of manufacture, it must be validated and the product should be well characterized, which involves a rigorous examination of its identity, that is, structure, physiochemical and immunological characteristics, then quantification of potency and purity, and finally the identification and quantification of the impurities. And however, for non-food and non-drug products, it is rather less costly, and the risk assessments, it will depend on the microorganisms involved, the specific product and the mode of its production and downstream processing. So we can say that this is obviously more complex for genetically modified microorganisms and pathogens. So as I was talking about the development of a pharmaceutical product, so from the initial discovery of the potential pharmaceutical, a patent application is filed. Then during this only means from the initial discovery to the product, between this process, product application is filed. Then which leads to the preclinical trials. Then preclinical trials is approved by the regulatory approval of human trials is taken. After the regulatory approval of human trials, clinical trials are conducted, and the average duration for the clinical trials is 6 years. And these clinical trials, they are divided into 3 phases. That is the phase 1, that for the healthy volunteers, phase 2 for small scale efficacy and safety testing on patients, and then phase 3 is for large scale efficacy and safety testing on patients. So now after the submission of the result of these clinical trials to the regulatory authorities for product approval. Now a product granted a license for manufacture and marketing. So a product is granted a license for the manufacturing and marketing. Now after granting a license, the product is available for sale. Then the trial for the phase 4 will come, that is the post-market surveillance. So once approved, the product must be manufactured to a specified quality standards at stipulated levels of purity and activity, which will depend on the individual product. So importantly, we say that the product, it must not be contaminated by potentially dangerous substances or pathogens. This means that a product such as an injectable drug for human use would have a somewhat different and more stringent set of quality criteria than silage for feeding to farm animals. Although these quality standards, they should be same whether for a human use or for any farm animals, feeding for farm animals, but they will be more stringent in case of human use, although the quality criteria will not be compromised with any of the processes. So now, talking about good manufacturing process and practices, like good manufacturing practices and regulation. So the FDA, it ensures the quality of drug products by carefully monitoring drug manufacturers' compliance with its current good manufacturing processes and regulations. Now this CGMP regulations for drugs, it contains minimum requirements for the methods, facilities and controls used in manufacturing, processing and packing of a drug product. The regulations make sure that a product is safe for use and that it has the ingredients and strength it claims to have. In the approval process for new and generic drug marketing applications, it includes a review of the manufacturer's compliance with the CGMPs. Then FDA assessors and inspectors, they determine whether the firm has the necessary facilities, equipment and ability to manufacture the drug intends to the market. So this was about the current good manufacturing practice regulations. Now, food safety regulation, it is controlled by 12 different federal agencies and derives their authority over food products from 35 different federal statutes. And US spends almost $1 billion annually on food safety regulations. So the federal agencies with some jurisdiction over food products, they are FDA, Food Development Authority, USDA, FTC, working for advertising, EPA for pesticides, CDC foodborne illness, US Customs for imports, and so on, DOT, NOAA, FS, SEFSAN, AMS, CVM, and we can see that they are different federal agencies working with different in different area. So now the approach to manufacture should involve an overall ethos of quality assurance, and this is defined as the sum of the organized arrangements that is made with the object of ensuring that product will be consistently manufactured to a quality appropriate to their intended use. And good manufacturing practices, that is GMP, they must be adopted within this. For example, those imposed by the US Food and Drug Administration, that is FDA, the GMP issued by the US Code of Federal Regulations on Design, Validation and Operation of the Pharmaceutical or Biotechnology Manufacturing Facilities. Now validating the standard operating procedures, that is SOP, it is also required. And their validation, they involves the establishing documented evidence which provides a high degree of assurance and that a specific process, it will consistently produce a product which meets the predetermined specifications and product attributes. So overall we can say that the GMP framework, it is responsible for the determination of the organization and functions of the production and purification processes and involve both manufacturing and quality control. So the quality control means it is concerned with sampling, specifications, testing, organization, documentation and product release procedures. So overall we can say that implementation of these practices, they should result in the controlled production of a safe product with the elimination of significant batch to batch variations in quality. And basically these now we will talk about the components of quality assurance, which I was talking about, the quality assurance. So quality assurance has 4 components, that is the product manufacture, product design, product development, research. And this product manufacture involves good manufacturing practices as well as the quality control. So now on the basis of the risk of microorganisms, World Health Organization they have classified World Health Organization has classified different microorganisms on the basis of hazards. So before any industrial process which can start operating, most countries they require that an assessment of risk analysis to be carried out. And this is specifically required for the process like genetically modified microorganisms. And risk is a function of estimated probability that an event will have an adverse effect. So now talking about, we can see in this table that these has been divided into 4 groups: risk group 1, risk group 2, risk group 3, and risk group 4, and the containment required. So risk group 1 microorganisms, the risk involved in group 1 microorganisms, they have low individual and community risk, and a microorganism that is unlikely to cause human disease or animal disease of veterinary importance. Then second, risk group 2. Risk group 2 include moderate individual risk, limited community risk, a pathogen that can cause human disease or animal disease, but it is unlikely to be a serious hazard to laboratory workers, the community, livestock, or the environment. So laboratory exposures may cause serious infection, but effective treatment and preventive measures, they are available and the risk of spread is limited. For example, salmonella, food poisoning, vaccines and antibiotics, and containment required is level 1 containment. Now talking about the risk 3, group 3, they have high individual risk, low community risk, and a pathogen that usually causes serious human disease but does not ordinarily spread from one infected individual to other. Then prophylaxis and treatment it might be available, and it has level 2 containment. Risk group 4, it covers high level and community risk, a pathogen that usually causes serious human or animal disease, and it may be readily transmitted from one individual to another, and no effective prophylaxis or treatment is available. For example, Ebola virus, but now we can say that this treatment of Ebola virus, but today's time we are talking about the COVID-19 coronavirus. So this type of disease, the pathogen which causes the serious human disease, it comes under risk 4, and even in today's time the vaccine is not, the treatment is not available for this corona infection, and it comes under level 3 containment. Now this figure it shows the category of process microorganisms and the level of containment required when used at research and industrial sites within the European Federation of Biotechnology. GILSP here means it is good industrial large scale practice, and GMM means genetically manipulated microorganisms. So here we can say the process microorganisms, that is the non-GMM and GMM, and this is the containment level required. So hazard group categories, as we have seen in previous table, that there are 4 types, that is 1, 2, 3 and 4, and here in GMM we can see the harmless group 1 and potentially harmful group 2. Potential harmless group 1, small scale, large scale, and it is A and equivalent, this is equivalent to GILSP, and this large scale to B and B1, where we can see that this is actually the category, these are the process of microorganisms which is depending upon the containment that is required. Now, safety precautions that is required for different levels of containment as required in the European Federation of Biotechnology, and the containment category as we have seen in this figure. So we can say that non-genetically modified microorganisms, they require GILSP 1, 2 and 3. Genetically modified microorganisms, procedures required written code of practice, biosafety manual, restricted access, medical surveillance, primary containment, use of closed system for viable microorganisms. And here we can see here that minimization of release, prevention of release, and this is treatment of exhaust gases also. Then the factors, removal of material products and effluents, penetration of the closed system, that's agitator shaft, monitoring devices, etc. And talking about the secondary containment, this was the primary containment. Talking about the secondary containment, it is related to the disinfection facility, emergency shower facility, airlock plus compulsory shower, decontaminated effluents, HEPA filters in air ducts, and area hermetically sealable, control negative pressure. So this is basically the safety precautions that is required for different level of containment. So now this was actually all about the regulations and safety process. And overall, we can say that the basically we can say that there should be a proper uh, regulation and safety for any process, and basically talking about the environment of any production system, the employees, they should be well trained for working in any process or and working under any process or industry. So this is all about product development, regulation and safety.